We are discussing complement system. We are going to continue this discussion of classical pathway. We previously saw how C3 can decorate the surface of a pathogen, which allows the phagocytic cells to recognize C3 and engulf it by a receptor-mediated endocytosis. So let's see what else our complement system classical pathway can do. Cleavage of C5. So some of the C3 binds to C5 and it causes a conformational change in C5, and that conformational change can be cleaved by C4b and 2b, which is present on the surface of a pathogen that we have already talked about. So which is now we can call this not only, previously we had said it is a C3 convertase complex. Now we can also call it C3 and C5 convertase complex. Cleavage of C5 by C3 C5 convertase initiates assembly of set of component proteins that basically make up the membrane attack complex. Basically, one strategy is to allow the phagocytic cells to engulf the pathogen. The other strategy our complement system is going to do is it is going to activate proteins which are going to make little holes on the surface of pathogen and causing that particular pathogen to die because of influx of water. Water will move into the pathogen and it will destroy it. The membrane attack complex can also be formed by another C5 convertase produced by alternative pathway, which we will talk about subsequently. But here let's see how this membrane attack complex is assembled. So we have talked about that C5 gets cleaved. First of all, C3 will bind C5, cause a conformational change. C2b and C4b will cleave the C5 molecule into C5a, which will diffuse off. And I would like to mention that this C5a is also an nephrotoxin, which will cause the release of histamine and the other effects of histamine release. C5b, in association with other proteins called C6, C7, and also C8, first initially these three proteins and then also C8 will join and they will get embedded in the plasma membrane of a particular pathogen. So this is going to prepare this particular pathogen for formation of little holes or pores that I talked about. So let's see what happens next. So here is the complete picture. So C5b associated with C6, C7 and C8 will form the structure, and it is going to be embedded in the plasma membrane on the surface of the pathogen. Now what this complex protein complex is going to do, it is going to recruit C9, actually 11 molecules of C9, and as you can see the C9 looks like a stick. So when you insert 18 molecules of C9, they will be actually in a conformation that is going to form a hole. As you can see, the hole has been formed. Water can enter, other ions can also enter the bacterial cell, and it can cause lysis of this particular cell. So this is the other strategy that our immune system, complement system can do to destroy a particular target. Here you can see an electron micrograph showing holes punched in the wall of a particular bacterium by the complement system. So we have just talked about two strategies our complement system has developed to neutralize a particular pathogen.